My name is Empress Williams. I'm a third-year biochemistry student, and today I'll be explaining my research on the exploration of the antimicrobial properties of Turnera diffusa and identification of its bioactive compounds. And so just some general background, natural products include plants, fungi, and other biotic material. Plant natural products, or botanicals, due to their unique phytochemistry, have played a major role in modern medicine and drug discovery due to increasing antibiotic resistance and their ability to combat and reduce the spread of highly resistant bacteria. The plant of interest for my research is Turnera diffusa, or damiana. Turnera diffusa is part of the Passifloraceae family and native to Mexico, Central and South America, the Caribbean, and Southern United States. Historically, it's been used as an anti-asthmatic, hallucinogen, stimulant, and a general wellness tonic. Our bacteria focus is MRSA. So why focus on MRSA in the first place? So Staphylococcus aureus is a very common gram-positive skin bacteria that can lead to serious and possibly fatal skin infections. Staph infections have become increasingly more difficult to treat due to their development of resistance to antibiotics. So Methicillin-Resistant Staphylococcus aureus is among one of many drug-resistant bacteria becoming increasingly more difficult to treat with contemporary medicine. MRSA accounts for over 70,000 infections per year and over 9,000 mortalities per year, according to the CDC. And with MRSA being an antibiotic-resistant bacteria and highly transmissible, it serves as an optimal candidate for research. So why explore Turnera diffusa in the first place? Well, as mentioned before, historically, Turnera diffusa was used as a general wellness tonic, but the Turnera genus has shown significant bioactivity. And this includes anti-anxiety, antibacterial, anti-diabetic, and antioxidant properties, to name a few of them. Another reason why I'm exploring this plant is because it contains compounds of lactic acid, quercetin, 3-O-gentiobiocide, betaine, and pinosembrine, which literature supports having potential antimicrobial activity. Another reason why we are exploring this plant is because a very similar species called Turnera omophila, or yellow alder, is used for pain relief, treatment for swelling and fever, which are all symptoms of MRSA, as well as a recent study finding that Turnera omophila is potentially active against MRSA. And so the question that we're asking is, does Turnera diffusa yield antimicrobial properties against MRSA? We began our project by submerging pre-ground plant material in methanol for 24 hours. The material was then drained and resubmerged in methanol before being extracted into 4 layers of methanol, hexane, ethyl acetate, and saline. Each layer was then dried down to later be tested for antimicrobial activity. And so both MRSA and Staph aureus were grown on an agar plate, and colonies were inoculated and submerged in tryptic soy broth to form a seed culture. The seed culture was incubated for 24 hours at 37 degrees Celsius, and then diluted with more tryptic soy broth, or TSB, for optimal bacterial growth. Biological assessments were performed with the samples on a 96-well plate, with the samples concentrations being at 110 microliters per mil. Then a spectrophotometer was used to generate the optical density of the bacteria, with the sample dilutions to measure the level of antimicrobial activity. And then ultra performance liquid chromatography coupled with mass spec was used to investigate the chemical composition of our Turnera diffusa samples, which will be highlighted in later slides. This slide shows a bar graph of the biological assessment of Turnera diffusa leaves partitioned in layers against Staph aureus. The x-axis represents the solvent layers, and the y-axis represents bacterial growth measured in OD 600, or optical density at a wavelength of 600 nanometers. And so the greater the OD 600 value, the more bacteria grew, which can be represented by the taller bars, and the lower the bars, the less bacteria that grew. And so Staph aureus was used for this biological assessment, which was a less potent strain. And this was used first to test against all of our layers. Levofloxacin, a known antibiotic, served as our positive control, and the vehicle, dimethyl sulfoxide, or DMSO, was the negative control. And our goal was to use these partition layers of Turnera diffusa to determine if any of the solvent layers displayed antimicrobial activity. There was little to no activity for the hexane and methanol layers, but there was a significant amount of activity based on the OD600 value for ethyl acetate at the concentration of 100 micrograms per mil. Once we observed there to be significant activity for the ethyl acetate layer, we moved forward and then tested it against MRSA in a future biological assessment. So based on the results from the previous biological assessment, flash chromatography was performed to collect fractions of the Turnera diffusa ethyl acetate layer to further investigate its biochemical components. A chemical gradient of hexane, chloroform, and methanol was used to separate and help purify the mixture. 6 total fractions were yielded based on the separation of the peaks, and a bioassay was performed to test the antimicrobial activity of each of the fractionated layers. So this slide shows a biological assessment of the starting material and the fractions being tested against MRSA. And on the x-axis, we have our fractionated layers and our starting materials at concentrations of 100 micrograms per mil and 10 micrograms per mil. And the y-axis shows bacterial growth measured in OD600. And so the fractions were tested, and most fractions had very little activity with the exception of fraction 4, which had significant activity at the 100 microgram per mil concentration. And if you look at the OD600 value, fraction 4 nearly completely inhibited the growth of MRSA, which will be the focus of our future studies to determine which compounds are responsible for this activity. So a common limitation to performing compound isolations is insufficient material. And after we collected our fractions of Turnera diffusa, we didn't have enough recovered material to isolate potential bioactive compounds. So we decided to scale up our extraction partition using about 800 grams of Turnera diffusa plant material. But in order to do this, we had to combine 2 separate batches of Turnera diffusa, one with pre-ground plant material and then one with a powdered form of the dried plant. And so in order to do this, we had to analyze both batches to confirm that they were in fact the same plant. And so we used a general mass spec method in positive ion mode and compared the base peaks of each sample. And so the samples were run in triplicates. So there are 3 of each, and at the very bottom you can see there's batches 1A through C, and then batches 2A through C. And the x-axis represents the retention time, and the y-axis represents the base peak intensity. And so the masses highlighted in green have a higher base intensities and allow us to confirm through our metabolomics analysis that the 2 batches were consistent with their chemical profiles when we compared them. And then based on the reported compounds of interest, the isolations will need to be performed in the future to identify and confirm the presence of these active compounds. And so a common limitation to performing compound isolations is insufficient material. And after we collected our fractions of Turnera diffusa, we didn't have enough recovered material to isolate potential bioactive compounds. So we decided to scale up our extraction and partition using about 800 grams of Turnera diffusa. And in order to do this, we combined 2 batches of Turnera diffusa, one with a pre-ground plant material and then one with a pre-powdered plant material. To do this, we had to compare the chemical profiles by performing a metabolomics analysis. And so we used a generalized mass spec method in positive ion mode and compared the base peaks of each of the samples. And so the samples were run in triplicates, 3 of each. And so we see there's a batch 1A through C and then a batch 2A through C. And then on the x-axis, the retention time is represented, and on the y-axis, the base peak intensity is measured. This is showing the stacked base peak chromatograms of each of the samples. And the masses highlighted in green have higher base intensities and allowed us to confirm through our metabolomics analysis that the two batches were consistent in their chemical profiles. Based on the reported compounds of interest in Turnera diffusa, isolations will need to be performed to identify and confirm their presence. And so some conclusions that we made through this experiment was that Turnera diffusa leaf extracts are active against MRSA. And in the future, further biochemical analysis will be done to identify and quantify active compounds. And then identified compounds will be screened against MRSA for antimicrobial activity. I wanted to give a huge thanks to Czech Lab at the University of North Carolina at Greensboro, as well as the Chemistry and Biochemistry Department. And I also wanted to thank the Undergraduate Research Scholarship and Creativity Office for allowing me to present at this expo. Thank you guys so much. Have a great day.